First of all, thanks for the invitation and kind introduction. So we've heard today a lot about sequencing, about biopsies, liquid biopsies, and I like now to focus on tissue biopsies and what else we can do with tissue samples beside sequencing. And I will focus on histology and present you our approach of an integrative diagnostic imaging approach. Integrative means in that context that we are able to spatially visualize entire tissue samples and afterwards perform sequencing of these samples. So what's the current workflow? How do we investigate tissue samples in pathology at the moment? So we have, for example, a malformation, some kind of tissue. Then we take a biopsy and then we can do sequencing. That's what we heard quite often today, but we can also do histopathology, histology. And first, before I will talk about our 3D histology approach, I briefly want to recapitulate how 2D histology or conventional histology works. And I think most of us are familiar to this process. So that means we have a tissue biopsy, we embed this tissue biopsy is then sliced into very, very thin slices, transferred to glass slides, and depending on the research question or on the diagnosis, we perform various types of staining for example, H&E staining or stain for specific markers for blood vessels, lymphatic markers, immune cells, etc. And the first part is a lot of manual work, right? So if you, for example, have a tissue sample, it's nearly impossible, or at least it takes a few weeks to section the entire specimen, to capture thousands of glass slides. And on the other hand, if you analyze these 2D glass slides, you do a 2D analysis of a three-dimensional sample. That means it's, this might be a bit difficult to understand the phenotype. And this is due to the fact that the missing spatial information is one of the major limitations of our classical 2D histology approach. And that's, for example, shown here and highlighted here on the left-hand side. For example, in the upper area, we see a 2D section or a 2D glass slide. We see some lumina, but only having this information is very difficult to understand a complex vessel architecture like in the 3D reality underneath our glass slides. In addition, we are interested, for example, in immune cells or in cell distribution. And if we analyze only 2D sections, this might be misleading. For example, in the middle, we see green and orange cells are represented equally. But 3D reality is different. We have more green cells than orange ones. Or if we want to detect rare cells, we might not hit it, that area, and therefore say, okay, there are none of these rare cells in our tissue. So that means we need more information. We need to analyze the entire sample. And in addition to this, we also have, by doing 2D histology, the problem that we have no technical replicates, that we cannot have a lack of resolution, meaning in the Z direction. And we always create tissue artifacts, tissue loss due to the physical sectioning. And we can, even use digital pathology approaches, but we do not overcome these limitations because they maybe count cells, they segment cells, but they do not solve the issue with the missing spatial information. And we all know that tissue homeostasis in disease is based on a complex interaction in three-dimensional space. And if we have, for example, like here on the left-hand side, this H&E image, we need something more. We need a third dimension to understand this in more detail. And that's, for example, here on the right-hand side now, a fluorescent H&E staining of an intestinal or of a gut biopsy. And by comparing both sides, you can easily realize how much information we obtain from 3D visualization compared to 2D. For example, we can see each single cell if we zoom in, or for example, if we combine this now with markers for lymphatic vessels, we can also visualize the entire lacteals in this biopsy. So this is then the approach, and we thought, okay, how can we achieve these 3D images? And we said, okay, let's develop a new approach we call 3D histopathology, or no touch, no waste histopathology. That means we have our sample, like always, it's formalin fixed, but then we do not do sectioning. We stain the entire sample, and then this sample is put into a light sheet microscope. Light sheet microscope, I always say, okay, you can compare to a CT scan, but you use, instead of X-ray, we use fluorescent light. And then you generate optical sections of the entire specimen, thousands of optical sections, and you can even detect each single cell in our specimen. And then you have all 2D sections. You can do 3D visualization, and like in an angio-CT, for example. And then you can use artificial intelligence to quantify, to segment structures of interest. And using this approach, we now get a much better understanding of the tissue microenvironment. And as it's a no touch, no waste approach, tissue is still available for downstream applications such as proteomics or NGS sequencing. So that's a short video from Zeiss. It's no advertisement, but I think it demonstrates the principle quite nicely. So that's, in this case, a zebrafish embryo. In our case, these are tissue biopsies, and now we are generating a sheet of light, or light sheet. And then we stack through the entire sample, like in CT scan. We generate thousands or hundreds of these 2D sections, and then can perform this 3D visualization. And that's a very, very fast approach. And we can, obtain images within minutes or seconds. And so these were some of our examples. So because lymphatic malformations can also happen in lung tissue, so we decided to visualize also lung samples. So these are healthy control lung biopsies, and we stained for fluorescent H&E staining. We did this kind of color adjustment because if you want to convince pathologists, you need to choose the right colors, because otherwise they will tell you, well, we are trained for 150 years seeing these pinkish blue structures. So don't take this away. And and what can we see? We can see that we detect each single cell in our 2D stack, and we could also see these pinkish structures, which are erythrocytes, for example, within the blood vessels, and also see all the lung-specific structures. And for example, such an image we generate in less than 1 minute. And this was an intestine 3D reconstruction I've shown you before. So we can now nicely see the entire intestinal histology, spatial histology, and can combine this, of course, also in the next step with IgG markers. Having said IgG, so that's our standard reagents at the moment to perform immunofluorescence staining of structures of interest. We can use IgG antibodies for blood vessels, for lymphatic vessels, for immune cells, for cancer cells, for whatever we want. And we have used these quite some time ago. This is an old video I showed 6 years ago, but I think it still demonstrates the principle really well. We see in white, we see blood vessels in the skin, biopsy, a healthy one. And in a few seconds, we also see in red the lymphatic anatomy. And using this approach, we now get a much better feeling and understanding of the vascular architecture. And once you have done this in controlled sample, then you can, of course, move on to diseased tissue samples. But if you use IgG antibodies, that might have some limitations, because on the one hand side, tissue penetration correlates with molecular size or molecule size. That means IgG antibodies are very big. Therefore, they penetrate tissue very slowly or do not penetrate them at all. It can take up to 1 or 2 months until you have stained your entire sample. And then you realize, oh, concentration was not good. Then you have to start again. And especially if you're a PhD student, it might be not helpful to finish fast. And and in addition, there are only a very limited number of IgG antibodies which are working in whole mods. So they are not suitable for routine diagnostics, because if you have a patient and you want to perform a fast screening in pathology, you cannot tell the patient, yeah, okay, let's wait 2 months until we have the results. So, and therefore we came up with a solution which are nanobodies. Nanobodies are single domain antibodies derived from llamas or alpacas, and they are only 10% the size of IgG antibodies. And due to their small size, they can penetrate tissue much faster, meaning we can perform a hormone staining of an entire tissue biopsy within hours, yeah, instead of weeks or months. And so that's a procedure, how it works. So we immunize our llama, then we obtain some plasmids later on. And on these plasmids, there is the DNA sequencing, sequence of our nanobody. We can express it in E. coli, which is very cheap. We can then directly label our nanobodies using fluorophores and perform the staining and 3D visualization. And that was the first nanobody we generated. It's an ESM1 nanobody staining blood vessels. And in the skin biopsy, you can nicely see the architecture of the blood vessels in this nanobody. But of course, for blood vessels, for lymphatic vessels, there's another marker which is quite popular, and that's CD31. So we also generated a human CD31 nanobody to visualize blood and lymphatic vessels. And these are only 2D sections, but you can easily see by comparing the middle image, the cyan one, with the left one, which is the current gold standard in pathology, which is the currently used CD31 antibody. You see, that the nanobody is even superior, that the staining is more crisp, more specific. And we then use these nanobodies to stain also additional tissue samples. Like here, this is a sample from the intestine, and we can visualize blood vessels, immune cells, not immune cells, lymphatic vessels, and in these structures of interest. And of course, you do not want to analyze or visualize, you want to quantify it. And therefore, we have also developed algorithms to extract and segment structures of interest, which we can then later on use to quantify our structures and determine parameters we're interested in, like vessel branching, diameter, etc. And having said this, we can also visualize immune cells. We heard immune cells are important, like macrophages. Here we have visualized, for example, CD31 as CD8 positive immune cells in intestine sample. In white, we see the CD31 positive vessels, and now in blue, the immune cells, and can nicely understand the tissue microenvironment. Coming back to the patient and the disease tissue, so we started with the structure on the left-hand side, the control. Then we went on to stain our first lymphedema patient, and at that time we performed 2D histology. Based on 2D histology, we could not identify what's the cause of the swelling in this patient, but by having these 3D visualization, you can nicely see they are lymphatics. These vessels are luminized, but they are disconnected, they are disrupted, and that explains quite easily the phenotype. If you have disrupted lymphatics, there's no drainage. And on the other hand, you can also use it as kind of decision support, because, for example, in such a situation, it's quite unlikely that a drug like sirolimus will help to reconnect vessels, whereas on the right-hand side, if you have these kind of malformation, these chaotic vessel structures, it might be more suited than in the lymphedema patient. But I like to have to point out that we are currently also analyzing a huge number of primary lymphodemas samples together with St. George's, and they look different. So it's not a standard phenotype for primary lymphedema. So we see a lot of difference depending on the affected genes. So, and these are some additional stainings from our recent publication. So we had some port wine birthmarks in a patient with Klippel-Tremonti, for example. And what did we found? We found as you can see in D, we found dilated lymphatic vessels, which are filled with erythrocytes, because if you do dermatoscopy of the patient, you see, okay, these are these red structures, but they look more or less like lymphatic, not like blood vessels. And using this approach, we could confirm that these are lymphatic structures which are filled with erythrocytes. And what's quite interesting, that these dilated lymphatic-like vessels show altered expression of lymphatic markers. For example, as you can see in D with the white arrow, you see this structure or this vessel is POX1 positive, but it's negative for podoplanine. And so that was a quite interesting finding. This is 3D reconstruction. Again, you see in green the lymphatic vessels. Lymphatic vessels are also a bit disrupted. We see some fragments, but what we do not see in podoplanine directly underneath the epidermis, these cystic lesions, which are only POX1 positive. We had another patient, and also in this patient, you see these with port wine birthmark. You also see these altered expression of lymphatic markers within the vessel, like again, POX1 positive, podoplanine negative. And coming back to what I mentioned at the beginning, so we want to have an integrative approach because we want to get as much information as possible from one identical sample. And so we asked, can we do NGS sequencing, for example, after we've performed light sheet imaging of the identical sample? And the answer is yes, we can. But for that, it's important that we keep in mind what we need for NGS sequencing. We need we have to perform DNA, RNA isolation, of course, but we need specific quality of DNA and RNA, because otherwise you could not perform sequencing. So, and as we work with formalin fixed tissue, you always have to keep in mind there's cross-linking, the chemical modification due to the fixation method. And we started to perform, for example, here, standard PCR of our gene of interest, and compared on the left-hand side, snap frozen control samples with a formalin fixed tissue and a formalin fixed and optically cleared or BABB cleared tissue sample. And you can see that the bands look similar. Then we performed also qPCR, and then of course used a bioanalyzer to check the fragment length or the DNA length, and could show that we can even obtain long DNA fragments. Then again, we want to perform also or analyze also the RNA. If you want to do, I don't have to tell you, but if you want to perform RNA analysis, you need a specific length, a specific quality. So we determined the diff200 value, which means the percentage of RNA or mRNA molecules longer than 200 base pairs, and we have in that case a value of 39. That means it's definitely possible to perform sequencing from these samples. And we started at that time with the TrueSight oncology panel from Illumina, because that was somehow one of the gold standard panels in the pathology department. You analyze 500 genes, approximately 50 RNAs, and it worked really well. And we could obtain really good results, which were definitely comparable to the current procedures. And our next step, of course, is to use the same approach for exome sequencing or whole genome sequencing or total RNA-seq. And with this, I hope I could convince you that's important. That's a very interesting and powerful approach to obtain as much information as possible from one and identical sample. And second last slide. So just say, today we heard a lot about the field of oncology for rare diseases. So we use some drugs which are used in the field of oncology to treat our patient. And I think in this approach, it's maybe the other way around. So our rare diseases, our lymphatic malformation can be a role model for that integrative approach. But because why? Because if you think about tumor diagnostic, if you have a patient with a lung cancer or potential lung cancer, then you take a fine needle biopsy, send it to the pathologist. The pathologist has to decide, well, it's only a tiny amount of tissue. Do I do histology or do I do genetics? I need both, but tissue is not enough. So with this approach, we now have, after imaging, still the tissue available and can perform the genetic analysis. And last slide, so just showing a mouse brain, just to illustrate that this approach is, of course, not only possible for human tissues, but also for mouse tissues. If we have, for example, some mouse models, and here we stained CD31. So it's possible to visualize the entire vasculature in this mouse brain. And yeah, we are able to phenotype and understand what's going wrong or what's going right in the tissue samples. Okay, and with this, I'd like to close my presentation, and thanks for your attention.